Good afternoon, everyone. Welcome back to the afternoon session of the Day 3 Gyan course. This session is the first national expert talk of the Gyan course. We have amongst us Professor Daman Saluja as a speaker for this session. Before starting the session, may I request Professor Yasha Hasija, Local Coordinator Gyan and Head of the Department of Biotechnology, to welcome Professor Daman Saluja, Senior Professor, Dr. B.R. Ambedkar Center for Biomedical Research, University of Delhi. Professor Daman Saluja has been working as a senior professor at Medical Biotechnology Laboratory, Dr. B.R. Ambedkar Center for Biomedical Research, University of Delhi. She got her doctoral degree in 1986 from University of Delhi. Ma'am worked as a research scientist in various capacities from 1987 to 1990 at University of Delhi. She then joined New York University Medical Center, USA, where she worked as assistant research scientist at Department of Biochemistry. Later, she joined Department of Microbiology, NYU Medical Center, and worked as an assistant research scientist from 1993 to 1997. Dr. Saluja joined Dr. B.R. Ambedkar Center for Biomedical Research, University of Delhi, as reader in 1997 and later became professor in 2007. She was also director of the institute from 2006 to 2010. Professor Saluja has received several awards and scholarship, including National Science Talent Scholarship 1975-1984. She is recipient of prestigious INSA Young Scientist Medal and LSS Kumar Memorial Award in 1989 by Indian National Science Academy. On May 11, 2013, on Technology Day, she was awarded the Biotech Product and Process Development and Commercialization Award of 2013 by the Honorable President of India for her outstanding contribution in the development of novel method of beacon-based PCR diagnostic kit for Neisseria gonorrhea and Chlamydia trachomatis. She was selected for Corona Program Technology Transfer Course 2013 under Indo-US Science and Technology Forum. For the past 20 years, ma'am's lab has been working in the area of medical biotechnology. Her primary contribution is in the development of PCR-mediated diagnostics assays for sexually transmitted diseases. Currently, her lab is working on LAMP-based diagnosis of Mycobacterium tuberculosis. In collaboration with industry, Dr. Saluja has designed a low-cost handheld fluorescent reader. More recently, her group has developed low-cost novel method of qPCR for the detection and quantification of BCR-ABL transcript for chronic myeloid leukemia (CML) and for acute myeloid leukemia (AML), and are also looking into anti-leukemic activity of plant extracts. She is author of 10 patents, applications, and 1 Indian and 1 US patent has been granted. Her laboratory is also working at the regulation of gene expression in eukaryotes using SYN3B as a model system. Professor Daman Saluja has published more than 85 original research papers in international journals. She has produced 20 PhD students, and more than 50 students have done projects under her guidance. She has been invited to more than 40 national and international conferences. May I now invite ma'am to deliver her lecture on innovative tools for advancing point-of-care diagnosis.
Thank you for the nice introduction. And why I chose this, because I thought it was a bedside translational research. So some of the translational work that we are doing in our lab, I will be describing that, and some other work which is going in parallel these days. More recently, in the last 5 to 7 years, we have come out with many more techniques, and as she just mentioned, that handheld qPCR method and device was designed in collaboration with other industry. We coming from the university and basic sciences, we devised the assay and they devised the machines, so we put it together too for the for easy testing and as a point of care test for the society. So that is our ultimate aim. So what we were talking about is why exactly we need this point of care test. And the requirement demand has really increased in the past several years. The reason for that is, one, awareness has increased, so people want to get themselves tested. Secondly, even in developing countries and poor countries. And secondly, there are, if you look into the Indian scenario, there are a lot of distances. So you can't always go to the city and get the testing done. You require a lot of expertise for people to carry out PCR, real-time PCR, the assays, even biochemical assays, it is difficult to carry out in remote places or in smaller setups. And therefore, this is very, very important to develop point of care diagnostics. So when we look at what are these point of care diagnostics, they are basically an integration of both, in engineering, these are chemistry and microfluidics and microelectronics. And of course, you require back-end software and other hardware to run these devices. So what do we what do these qualify? How do we say this is a good point of care device? WHO says your any diagnostic assay should be robust, which means it should be rapid or quick, accurate, easy to use, portable, and low cost, and it should be repeatable assay. So, keeping that in mind, a large number of new devices are coming up, and you will actually, if you will see in the last 1 decade, lot of point of care devices have come up based on microfluidics and paper based microfluidics, whether it is chip based microfluidics, and they are now we are even connecting these devices with the phone software that you can be anywhere and you will get your results and all that. So, of course, in developing and poor countries, as I told you, it is very important because hospitals, one, are overcrowded because of high population. Then distance to reach these places, hospitals, is very challenging. Many of the smaller places have poor infrastructure, and often, in poor and developing countries, we have outbreaks. When there will be rainy season, then we will have one set of infections coming up. When it is winter, then another set of infections will come up. So it is always the problem with the society, in the society, to have, it is like a more like a challenge to able to give the right medication at the right time. And because of that, we lose the footfall. A patient, if is told that you come after 3 days to get the test report and then your treatment will start, he or she may not come to the same clinic. By then they might do their own other kind of decoctions and cocktails, or take these medications from over the counter, which has resulted in lot of antimicrobial resistance in the society. These days we are always talking about AMR and multi-drug resistance, and that has come up because of majorly the contributors are developing and poor countries, because the medicines are available on the counter. So this has really become a challenge, and therefore it is very important that we should reduce the time of diagnosis, so that while the patient is still in the clinic or in the hospital, we get the results and then we can start the treatment the same day. Okay. So keeping that in mind, I think the priority by the WHO was put that we must develop good point of care diagnostics, and they have to be approved by WHO or by some society like that in your own country, so that they can be, like in India there is NBL is there, which looks into the quality of any assay that is developed. Even only then it is approved for the use in the hospitals. Okay, so the requirement for any point of care is basically it should be reliable disease quantification, it should improve the follow up, and many of these new point of care diagnostics can even develop, they can give you results on the genetic sequencing in a short time. They are also, many of them are based on proteomics and genomics and transcriptomics data, and nowadays big data is being analyzed
to find even the new biomarkers, or to discovery of for the discovery of the drug discovery pipelines, so that we reduce repurposing of new drugs, lot of these which is going on in your department also in many labs. Okay, so the advantage of this will be, of course, you can stratify even a given disease into major subtypes. Like for example, I say, for example, sexually transmitted diseases, they are caused by more than 20 different pathogens, but some of those pathogens have developed antimicrobial resistance. If my diagnostics test is able to tell me exactly that, okay, this is in the patient is infected with X pathogen, and it is resistant to this antibiotic but susceptible to this antibiotic, or to common other one or two antibiotics, then it is very easy for the clinician to prescribe the drug. And that is what we are trying in mycobacterium, where we have a huge burden of multi-drug resistance. So now there are devices, I don't know how many of you have heard about Gene Expert, which uses the first line of drug that we use. It has the mutation in that results in the drug resistance, and that is used as a Foster indicator of AMR or MDR in case of tuberculosis. But we need to develop such methods for other diseases also, which are common, and especially in developing countries or in poor countries, because many of these AMR and many of these problems are less in the fully developed or industrialized countries, because they don't have that much problem of infection. But in India and other Africa, even Hong Kong, Bangkok, you will see lot of AMR is coming up, and that is creating lot of problem. So you can really, so even for others, now how many of you have seen glucometer? It is so convenient. A person having diabetes need not go, they can even monitor morning, evening their sugar, and those who are on insulin, they are able to do the test and then decide how many units of insulin they have to take. Okay, the life has become easier, it is better managed, diabetes control management has improved, and there is no inconvenience to the patient. They don't have to keep going to the clinic. So can we really bring such devices which are even useful at home? The patient themselves or somebody at home can run that device and bring, and you have all seen it is so easy. It's just pin prick, and then you put your drop on the strip, and it will give you within few seconds, it will give you your sugar levels, it will tell you. So similar device has now come for the hemoglobin determination. So it not only does, especially this is very useful for determining white blood cells, which is a Foster indicator of the infection also. And in many cases that is very, very important, especially in leukemic patients. So it can be done at home, nothing, same way you do a pin prick and then you can check. Everything is in the device. So when you put the chip that they have put, it's a more like, there are many versions of this. So even there is a kind of a paper strip kind of a thing, and that can lead to removal of some of the cells, and especially the red blood cells. And then there is a principle by which it determines the white blood cells. So such point of care devices are very much in need for many such diseases. Okay, so there are other point of care devices which are in making, not only determining the hemoglobin, RBC and WBC, but circulating tumor cells. Now from this white blood cells, people are also developing various versions, and some of them have come into the market. Little expensive, like they may be like under 50,000, but there are various versions of that. So depending upon the requirement of the patient, you can buy anywhere from 2,000 to 50,000 device, which is affordable in many cases. But now they want to even look for circulating tumor cell such devices. The patient may not want that at home, but if it is present at any local clinic, it is so easy. If there is a doctor has any suspect for the patient that there is some symptoms and he wants to check whether there are any circulating tumor cells, then this device can measure, and many of these, they basically capture these circulating tumor cells by antigen antibody interaction. So this function as an antigen, you have antibodies, and then there are magnetic nanoparticles which will concentrate it, and then you can measure it. So basically you require a microfiltration where only you will filter out the proteins and other RBCs, and then the principle of hydrodynamics will follow, and then under the various dielectrophoresis will be done, and where you have magnetic beads which are bound with the antigen. So if there are any antibodies you are looking for, you can look for antibodies, or if you are looking for any okay. So I thought there was another figure. So they will antigen antibody interaction will occur in the magnetic field. So beads will remain stationary, or you can put them in the microfluidics. You can channelize them into the microfluidic strips, and then measure by the spectrophotometric method. Either your antibodies are fluorescence tagged or they are enzyme tagged, and those substrate if you put, then they will give you the estimation. So these devices have become very, very useful, although they have not hit the market in developing countries, but they have been developed, and they will sooner or later they will come into this, and this will become a very good method. And in fact, the principle of this technique is what we hear these days, liquid biopsy. How many of you have heard about liquid biopsy? Anybody here? So liquid biopsy is becoming 4 or 5 years back, it had become a routine in USA, but now many hospitals in Delhi, at least I know, offer liquid biopsy, and the advantage of that is you are going to take the blood sample. Otherwise biopsy is very invasive. You have to, under ultrasound guidance, you cut the piece, then you isolate, you grow the culture, you look under microscopy, then you do immunohistochemistry to see what kind of antibodies are present, are there tumor antigens are present or not. So it is a long process, and normally 72 to 96 hours they ask for giving the report. Okay, there is one is FNAC, fine needle aspiration, and one is biopsy. So liquid biopsy makes it very convenient because all you have to do is, it is far less invasive because you are taking blood samples. People are more, comfortable giving blood samples than, getting anything scissored out of there, and sometimes some of the biopsies have to be done in the OT only. You can't do it outside also. So that way, this liquid biopsy has become really revolutionized the tumor detection. So some of these tumor cells dislocate and they come into the bloodstream, and you are looking into those cells which have come in the bloodstream. But obviously the number will be less, right? So you have to devise a method to concentrate them, to get rid of other kind of molecules, but you should only have those cells which are expressing the antibody, tumor antibody, or outside, or tumor antigen on the surface, depending upon which type of cancer you are looking up. If you are looking for any receptor, then the cell will have antigen on the surface, and you use magnetic beads which will have antibody bound to them. But if there are any kind of tumor antibodies are being produced, the body also has some defense mechanism. Then you look for the antigen, whether this antibody is binding to that. But in liquid biopsy, mainly it is the antigen that is looked, and the magnetic beads and the antibodies are put into that, and then you wash away all the so you do carry out this microfiltration, and hydrodynamic properties will separate the some of the cells and from the others, and then the run it on the this on this is going to happen on the same device. After this filtration, the magnetic beads carrying those antibodies or antigens, they will move into the microfluidic channels, and then there they will be bound to the to antibodies, and other unbound antibodies will be washed away, and then you do the simple, the substrate is lying in the end, and it will give you a particular whatever color or color reaction it has to do. So this has become, yes.
So do you think the detection limit has to be so sometimes, and how often does it miss it? Like, what if there isn't enough? Or
Yes, you are very right. So this is like at the for the time being, as I understand, it is being used as a primary detection. Like earlier we used to do FNAC as a primary detection, and then do biopsy, and then histopathology. So and both FNAC and biopsy are highly invasive, and in some cases, like in bone marrow biopsy or GI tract biopsy, it is basically almost like a surgery. You have to give a minor cut to do the biopsy. Oral carcinoma biopsy, so there it is very good, because if you get positive result, you know it is positive, but if you get negative result, then you have to worry about whether you want to call the patient after another 15 days, 1 month again, or you want to do other secondary test and you let him go through the MRI.
rely on it, but at least
Yes, I think it has not come to that robust a condition, but they have already started, because at least you get rid of major, where there are more problem in this. For breast carcinoma, they are using it very, they say it is very accurate for breast carcinoma, because it's a very ductal organ. So the antigens, tumor antigens will move very fast to the lymphatic fluid and then to the bloodstream. So it is easy to catch them. And also we need to identify more tumor antigens, because if we have a battery of tumor antigens which we have identified, then we can have multiple antibodies. So chances of missing are much less. But currently we have very handful of antigens like CA123, CA125, and prostate cancer ke liye PSA is there. So we have very limited number of antigens that we are very sure they are good antigens as a marker. But people are now looking a lot into biomarkers, and with big data analysis, transcriptomics and proteomics now, a lot of work is going on in that direction to identify the biomarkers, the genes, proteins that are definitely upregulated in certain types of cancer. And I am sure in another couple of years, we will have this, with the help of AI and machine learning, we will have very good biomarkers which will work across pan cancer, because many of those genes are overlapping in multiple cancers if you do by this transcriptomic data and you use AI facility. So I think we are not far away in identifying more good biomarkers which will be specific. So if we have 3 or 4 biomarkers, 3 or 4 antigens, and they turn out to be positive, we can have them different labels, different fluorescence tags also, and then it will be very easy to identify that yes, there is cancer. First problem is that. But if you look into cancer treatment, medication drugs are not too many. Many drugs are used for different cancers. Same drugs are used for different cancers. So I think at least first line of treatment can begin immediately. The early we diagnose and early we start the treatment, the patient has better chances of survival and recovery. Management is much better. So yes, your point is very correct that it is, at present we are still in a dual mode, research come final devices. So this is, I found one of the companies where they were doing many of these things. So they were taking, and I think the engineering students, or if they are here, they will be able to appreciate it much better. So these are the chips that you can see in this and in the figure A, where you have many of these. So I don't know if you can read on the background, but I could not do much about it. So there are cameras, there are microfluidic chip at the bottom. So the camera will move and see the capture the even the fluorescence or the cells, and whether they have the antigen antibody interaction is occurring, then there will be activity will be there. Okay. So these kind of it is not only that you are just doing positive, but if using these magnetic beads, if you are capturing these tumors cells which are expressing certain biomarkers on their membrane, then you can capture those cells, elute those cells from those microbeads and use them for RNA extraction, protein extraction, and carry out, these days you can even do with nanograms of RNA, you can do so many PCR reactions and stuff like that. So you can do any kind of downstream processing with those cells. Although this is more at the research level currently, but this is the thought that they are working with, and many laboratories and companies are working now to bring it up to a level where these beads will not only recognize the tumor cells, but then you can also concentrate, these concentrated beads can be utilized for isolation of RNA and the protein, so that we can really come out with the more and if it is an infection, we can also identify the microbes. In this direction, microbes, actually some more work has been done, and some of the laboratories are actually purifying these microbes from the sample, and by using antibodies against the membrane proteins, and then using that for culture and for the other things. So that is little bit more working in a much better direction. But for cancer, of course, we are still little bit lagging behind and not up to that. So then what do we do after that? Once we have identified these tumor cells, or we have purified these cells, whether they are carrying microbe or anything, then we have to identify things, right? So identification can be done, of course, with proteomics, which requires little more sample, but PCR is one that can be very, very easily done. Of course, you can do PCR directly also. So and because now you have seen during COVID, there have been many point of care devices for the PCR. So PCR is much easier, can be easily used, as well as real-time PCR has now become very, very common. So I will first briefly introduce you to people to the PCR. I am sure some of you are very well versed with PCR, but those who are not versed, not familiar with PCR. So this is a nucleic acid amplification technique in simple terms. Okay, so like we use amplifier in music system, this is a specific part of the genome you can amplify to whatever extent you want, it will amplify it. Okay, so how it will do the specific ones is by the specific oligonucleotides that we use. Now it was just little bit of history, like Carimules, in fact he was on a recreation trip, and that's when he saw the hot spring. It's a very interesting thing, you should see how mind works. So he thought these organisms are also multiplying. that means they are having DNA replication, but their DNA polymerase is very stable because it can handle such high temperatures. And how they must be, replicating, it was like out of curiosity, he took a sample of those these thermophilic microorganisms, and isolated DNA polymerase from them, and found the peak, the activity peak or the maximum activity comes in the range of 70, 65, 70, and like you see a bell-shaped curve normally for any enzymatic reaction. So that's when he discovered PCR. So he took it that if you isolate DNA and denature it at high temperature and allow it to cool and use this enzyme at 65 to 70 degrees, then it will result in the DNA amplification. Now also remember, if you have read in your BSc and MSc, DNA replication always require primer, not RNA transcription. So it cannot, DNA cannot start ab initio. It requires a free hydroxyl group at the 3' end, to which the next base will be added. So what he did was he synthesized the primers for the desired gene and then did this in a test tube. So you will be surprised what you see today, the PCR machines. When he did his first PCR that he standardized, it was 3 water baths. So he would denature at 94 degrees, quickly shift the tube to the 72 degrees, put the enzyme, and then go to allow it to anneal and then to the reaction. So this he would do for 30 to 40 cycles, and he found huge amount of specific DNA fragment. Okay, so this was very interesting, and that revolutionized the whole molecular biology or biotechnology, whatever you want to say, because now we could amplify the desired genes and we could use them for studying their function, their structure of the proteins, and whatever we wanted to do, kind of mutations that it has in the organism, or we want to modify it, how it happens, what is happening, everything you could study. And that was very interesting, and that led to the applications of PCR, led to the Nobel Prize given to Kerry Muller and Smith. Okay, so this is what I told you, that you have primers complementary to the 2 strands, forward primer and the reverse primer, and this the synthesis will occur only between the 2 primer lengths, and it will go. And now, if you look into, all you are adding in this PCR reaction is your DNA, your primers, DNTPs, you just throw DNTPs, buffer, and that's about it. Nothing else, because all your buffer contains the divalent magnesium ion and all that, and DNA polymerase. So earlier it was used DNA polymerase of these, and now we call it Taq polymerase, or because from lot of other organisms were identified, they all had thermophilic, from the thermophilic spring, hot spring, so they were also used, and now we have several polymerases that we can use, Vent polymerase and Taq polymerase, and now they are cloned, their 3' exonuclease activity, 5' exonuclease activity has been deleted, and lot of manipulation has been done with the enzyme, so that you get better processivity and you get better results. Okay. Any questions so far? Any question? Good. Am I going too fast? No. Okay. Fine. All right. So this is something, is the basic PCR amplification. So you have one template. From the first cycle you form 2, say you get 2 raised to the power 2, you form 4, then 2 raised to the power 3, you form 16, 32, and by the time you reach to the 35 cycle, theoretically you have 68 billion copies, although that will not happen because some or the other agent will start becoming limiting, so that is, but you still get ample amount to do any downstream this. And then once this was worked out, people started looking for more applications of PCR and many changes were done. This is just the same thing that you denature at 94 and then you allow it to anneal, the primers are allowed to anneal, and then you add DNTPs and you have the extension. But now we have automatic machines which will result in changing these temperatures. So each cycle has a denaturation step, annealing temperature and extension. They have come up with many more things where you can do your whole reaction at with two temperatures, 94 and 62 or 65, where there will be annealing will also occur at the same temperature and extension will also occur at the same temperature. And the idea is you make primers such that their TM is also in that range, and also the enzyme has been made in such a way that it can work at 60-62 degrees with full efficiency. Okay. So, but the main trick lies here in the primer designing. So you can develop any diagnostic if you have very specific primers. Okay. So how you decide on the specificity of the primers? One is that the primer should not hybridize to any other site in the genome of the microorganism. Now, when I take a patient sample and I am looking for microbe A, I don't know what all other microbes they have in their sample. Okay. Now, the problem comes is because we do not know all the microbes, we only know a handful of microbes whose genomes have been sequenced. Like there are billions of microbes in over there. I'm not saying all of us have billions of microbes in each infection, but even if there are 100 microbes or even 20 microbes, I don't know which 20 are there. So my primers have to be specific. Now when I say my primers have to be very specific, I can only hybridize with the known genome sequences and see my primer does not bind to any of these microbes. So you have to experimentally check that your microbes are specific. How will I do that? If my organism gives a amplicon of about 220 bases, then when I do it with any other microbes for 30 different patient samples, I should, and they are known to carry that infection, I should, from all these patients, I should get only single band of 220 bases. So you initially, when you are developing a diagnostic assay, you develop the specific primers, you standardize the conditions, and then you always look for patients which are already known to carry that infection. This is not a blind assay, this is developing, you are in the developing phase. Once you have proven that your primers are specific, then you start doing blind assays, single blind assays that one person does with a standard gold standard protocol and one person does it with the new method. They don't know the results for the same sample, so it is blind for you. So you are given certain numbers. They know which one is positive and negative by their gold standard, and you have to see whether your results tally with their results or not. Okay. And then you do double blind. Double blind is that you take the samples and they also don't know at present. They also do their methodology, you also do your methodology, and if there is any discrepancy that they are getting positive and you are getting negative, then you use a third test to confirm whether it matches with your test or with their test. This is called discrepant analysis. Okay, and there are certain mathematical derivations which you can use to check the sensitivity and the specificity of the reaction, predictive positive value, predictive negative value. How that predictive positive value is when you say a positive test will always be counted as positive, and predictive negative value is a negative test will always score as negative. So that has to be the best. Okay, if it is 100% PPV and 100% NPV, then your assay is perfect. But nothing is perfect. But WHO takes it that if it is more than 95%, then it passes their test. So any test which comes in the market has to be approved by, like in India we have CDSCO, and in outside in USA it is FDA approval, and then overall if you get WHO approval of your assay, then you can market it in any country. So this is very, very important, and the specificity always come from this. And also interestingly, if I have very short primers, I have more chances of non-specific binding. But I should not have very long primers either, because then also I will have non-specific hybridization. So even if if I'm using a 30 base pair primer and 4 have not bound, it does not matter. Even then at 60 degree it will remain hybridized, right? So that is a problem. So you have to find, should not be more than 18 to 20 mers, it is good enough. So it should give you a range of this thing, and normally, like we choose 1/4 chance of finding A, G, C, T in any, and 1/16 of this. So we try to look into almost number of G's and C's, and A and T should be approximately the same, so that your TM is around 60. That is what is important. Okay. So, but for some cases we also use universal primers when we don't know any pathogen is present or not, new pathogen, then we use universal primers. Universal primers are ribosomal gene primers, 16S ribosomal for microbes, because ribosomal gene is highly conserved among microbes. And then if I want to look into all the species of a genus, then also I can use certain primers which are specific, some protein which is specific to that genus. I can design primers against that, and then it will give me the how many of them are present. The length, the size of the amplicon may be variable, and that we can use. But then specific is when you are amplifying a single sequence from a milieu which may contains hundreds of other microbes. That is your specific test. All right. So forward and reverse sequence, that I have already told you. And Taq polymerase, because it is Taq is from Thermus Aquatics, but there are other which have now come, and this enzyme was the first one to be utilized. So, and component of PCR I have already told you, template and primers, and then the buffer and this thing. So the melting temperature is the most important thing that you have to keep in mind, because melting temperature TM should be around 60, between 60 and 65, not less than that, and beyond that. You can also calculate it by this formula, 4 into G plus C plus 2 into 4 plus T, but these days there are many softwares which will give you the TM, and you must make sure that there are no hairpin loop like structure, and also the forward and reverse primer should not hybridize to each other. They should not form dimers, because they are small molecules. Then they will form dimers, they will never give you amplification, you will have poor amplification. Yes.
The sequence for which we are making primers against, if just for the sake of the complementarity, it has the hairpin loop structure. How do we synthesize?
So you can always choose nearby area. You can, for diagnostic purpose, that is not a problem, because you can always make, Instead of getting an amplicon of 200 bases, you may get an amplicon of 210 or 190, because you can little bit move your primer upstream or downstream on the length. Problem comes when you have to do cloning, because you want full length protein, right? Or you want to clone a subdomain, then there is less problem. But if you want to clone a full length protein, then you have to be very careful. You can't leave N-terminal region for sure, but you can always take little bit extra 3' region, because वहाँ तो termination codon will come, so you will be fine, right? So that is the main problem where you have to take care of that. There are ways to circumvent that. You can put certain, you remember the wobble hypothesis, right? So the third base can be slightly different, so you can make primer like that, but then the primer should not end there. So I can make a primer which is little longer going into the gene, but that base I can change. So it is like the same principle of site-directed mutagenesis that we use. So at the other end, if there is a change in the base, then there will be less of problem, because the last 3 or 4 bases should bind perfectly. Otherwise, you will have non-specific hybridization.
So we are allowed to induce some kind of gaps, not essentially gaps.
But you will not worry about that, because your amino acid will be the same because of the wobble hypothesis, right? When we do site-directed mutagenesis, then we make a change. It should not be the same amino acid, but here you can always do that. So there are, like, thumb rules, but then variations are always there. All right. So secondary structure should not be formed, and this is what you have. We are coming back to the same slide. Am I okay? So buffers and thermocycler. This is the most common PCR machine looking like these days. They, instead of having 2 blocks, they have even 3 blocks, by which you can run 3 different PCR reactions independent of each other. Independent time पे डाल सकते हो, and you can set the annealing temperature and extension and whatever you want. You can program them separately also, okay? And you can store your program, so whenever you are doing next time, you need not worry about again looking into the programming, okay? So normally this is what we do, and you normally end up doing 30 to 35 cycles. The only problem with PCR is you only see at the end of the reaction, and it is not quantitative, right? So you run it on the agarose gel, you see a dark band, and then you say, okay, this is more or this is less, but you don't know how much more or how much less. Hey, okay, it is double or maybe little more than double, but it is not truly quantitative, okay? So it is very approximate, but yes, if you do not get in 30 cycles also any band, then you know it is absent. That particular microorganism is absent, okay? So lot of changes also came up, as you will realize. If I want to do site-directed mutagenesis, or if I want to look into a mutation, especially for AMR, antimicrobial resistance, then I need to design primers which are going to be just looking at that. So in the culture or in the sample, you will have some bacteria which are having a wild type sequence, and there will be some bacteria which will have a mutant. Can you tell them just to be little less louder? थोड़ा धीरे बोलिए, बच्चे डिस्टर्ब हो रहे हैं. So then you have 2 types of templates. So you want to make sure that both the templates get amplified, and you are able to distinguish the wild type from the mutant one. So many new deviations have come up. So you can use this as a you can do nested PCR for that. Hot start PCR is only to improve the this denaturation of the template DNA if it is very GC rich. So that is used. Then there are sometimes you want to do long PCR, so there are separate enzymes which have better prognosis. And then there it comes, the nested PCR is mainly used to look for specific mutations that you want to see in the background of even your wild type. And then of course you want to look into the gene expression, so you have RNA. So once we identified these and sequenced it, we knew where exactly is the splice form. We did Tachman polymerase strategy, and we have quantitatively shown that how in oral carcinoma stage 1, stage 2, stage 3 the splice forms vary. But this is the advantage of doing the PCR, and then you can always couple with real time PCR. It is not always that you have to do, to begin with only you have to do real time PCR. Then another very powerful variation of PCR is restriction fragment length polymorphism. Right, you will get the this thing. But many a times your DNA is in very limited condition. So what they do is they amplify first and then they do restriction digestion. Now if there are mutations in the gene, then what will happen is if you do the PCR around that, so you will get and do a digestion with the known restriction enzyme. If you are lucky that your mutation falls on the mutation in the resistant site where it cannot be digested by the restriction enzyme. So, but you can use this in many other devices, like here it has been shown. I think this was for a here they have done after PCR, or they have done southern blotting, but you can also do it by simple RFLP. So after PCR they did digestion of the whole genome, and then they looked at the probe. So the next experiment can be a probe based, or you can do simply by restriction enzyme. But here, because there were so many bands, they tried to do it by the probe. And RFLP analysis is also used for like sickle cell anemia, and there you have two alleles. One is beta A allele and beta S allele, which is sickle cell anemia allele, and there is a MST2 restriction site is present, and you can see that where the base is changing. CCT instead of G, you can have any other nucleotide, and this result in loss of the MST restriction site. If there is a change in this GAG sequence, but it has to be, AG has to be there. So if you have in the beta sickle cell anemia, it is CTG instead of GTG instead of GAG or any base. If the G would have mutated, nothing would have happened. But so what happens is, if you take the patient to sickle cell anemia, if you do pedigree analysis, you will see that a person which is carrier of the sickle cell anemia, that means because you always have two alleles are present. So if there is one is normal and one is sickle cell allele, you will get 3 bands. One will not be digested with MST, and the other which is digested with MST, you will get 2 bands because you have amplified that region. Then both the parents have the are carrier, and now the offsprings you will have combination. One will be only AA, which is normal, so you will only get smaller bands. One which is sickle cell, which has mutated, you will see the larger band, and again one is who is carrier will have 3 bands. So you can very easily distinguish which child is going to develop sickle cell anemia, right? And even for antimicrobial resistance, you can do, as I told you, for ciprofloxacin resistance. How many of you must have taken fluoroquinones like norfloxacin? And so earlier, 10 years back, ciprofloxacin was the one which was most common drug. But bacteria are also very smart. They mutated. So now Ciprofloxacin cannot function in these bacteria. But there is a very simple way to check by PCR followed by RFLP, restriction fragment length polymorphism.
These
Yeah.
spots here, like for the mutated DS, here we see only 2 sites and here it corresponds to 3. So are these the MST2 cut sites or these are?
Yeah. So these blue ones are MST cut site. So if I have amplified this region, you can see a blue line. Okay. So you should get this fragment in the, you will get only the large fragment in the sickle cell anemia. It will not get digested with MST. But due to mutation, because GAG site has been converted into GTG site, then you have lost MST. But if you amplify the same from a normal person who is not sickle cell anemic person, then MST will cut in between also. So you will get one fragment will be this and one will be that, right? So you will get 2 fragments. Now if a person is heterozygous, you will get 3 bands because 2 will come from the normal and healthy allele and one will come, a larger band will come from the mutated allele. So you will see 3. So all 3 are carriers, but if you get only one which is larger, then it is completely sickle cell. If you get only these 2 smaller one, that means they are carrier for both alleles, they are normal alleles.
Okay, got it.
Okay. Similarly, you can use it for ciprofloxacin resistance also, and for many other cases we use this as a marker for. But only thing is that there has to be a restriction site in between that region. Then only it is possible, and the mutation must occur overlapping the restriction site. So there are too many ifs. So that is why it is not that common. But otherwise you can use RFLP analysis in this, like for example I had shown you. So you will get from one person to another, the profile will be different, and this is very important in your paternity dispute that we hear. So they do VNTRs, they make the primers for VNTRs and amplify the region in between. So variable number of tandem repeats will help you decide the PCR profile. Multiple bands will come, and if you match the multiple, I thought I had put, let me see, yeah. So you can have it even at the criminal side, you can have it paternity dispute. So one person will have, so if it is like, let's say I will explain you for the crime side, the data is there, then you can also extrapolate it for the paternity dispute. So the survivor, you took the swab from the survivor and carried out the PCR using VNTR's primer, so you got a pattern. Now you compare the same set of primers you use for this, and this is the biological sample from the crime site. So crime site primers, there are many bands which are matching with the survivor and then with the criminal. So whichever is there which is matching with this crime scene, that is the person which is. Can any one of you tell me who can be the criminal out of 1, 2, 3? 3, because their bands are matching from the biological sample at the crime scene. Okay, so this is being used. Then many a times what happens is that in the nails, when the person is trying to save himself or herself, then there will be some ruffling between the killer and the person. Then it will happen. In rape case, semen, sometimes they are able to get semen from there, and there they are able to identify the pattern. So this is same thing happens in the paternity dispute. There the child, whatever bands child has, they are either inherited from mother and father. So the one will be their true parents and the one person who is, there is a dispute, they may or may not be the true parent. So there are two sets of parents. So they will do the PCR from all the four. The child band should match, all the bands that they get should match with either of the two parents, like either the mother and the father. But if there are certain bands which are not matching with either of the father or mother, then they know they are not their true father and mother. Okay, so that way paternity disputes can be, this is a very strong evidence which is now even accepted in the court as an evidence. It can be utilized for the search.
One question. The biological evidence at the crime site, we are not sure that it's only of one individual. There could be multiple DNAs present.
Right, you are very right.
So how do we do that?
So in that case, there can be, if there are like that happens,
But mostly there will be multiple because one of the killer or maybe the attacker and one of
no,
the
somewhere at least one or the other suspect they will get, because they take, it is not that they will take sample from only one place. There will be multiple sampling is done. Supposing let's have a scenario that 3-4 people came to kill a person, right? So somewhere or the other they will, all the three will leave their mark. Supposing in the ruffling somebody has bled, one person had some bleeding, so uska kahin na kahin blood drop gir jayega. Or even if they are able to catch one person out of 3, कहीं पे उसने अपने आप को बचाने की कोशिश करी, बाइट कर दिया, हेयर फॉल हो गए, बाल खींचे, हाथों के नाखून में आ गया कुछ, तो all these things are taken, and then even if one suspect will be catched, then वो ही बता देगा बाकी के कौन से लोग हैं. But many a times they are able to detect more than one from these evidences. Some problem does come in rape cases where there are multiple people are there. So, but there also somehow they are able to decipher the suspects. I think there also they take multiple sampling, and somewhere or the other if they are able to get 1 or 2 people very cleanly, then they are able to catch a rest of the people from that. The person will tell even this was involved, because they want to now escape. They want to either divide their fault or whatever. Okay, but कई लोग जा भी बच भी सकते हैं. So genome mapping के लिए we use RFLP, localization of genes for genetic disorder, risk for the disease, paternity testing, genotyping tests, and forensic sciences for suspects. Okay. Now this is basically, you can also do real-time PCR rather than doing RFLP after PCR. And here also you can do multiplexing PCR. Like this is an example from our work. You can see that with increasing template concentration, you will get lower and lower CT value, which is, all of you are familiar with the CT value after COVID. So that means it will, the graph, there is a baseline and the graph will start rising from there. So higher the concentration, more will be the, so if there are 10 raised to the power 4 templates, then it will come up later than when if it is 10 times more or 100 times more. So CT value will keep on decreasing. Now this becomes quantitative, and this was important in the COVID case. They used to say if your CT value is more than 30, you are almost cured. But if your CT value is in the range of 17 to 25, then you still have very active infection, right? That means you have lot of pathogen in your bloodstream or swab sample, nasal swab or pharyngeal swab, and that large amount of pathogen will give large amount of RNA. आपने तो सेम वॉल्यूम में उसको डिसॉल्व करा, कहीं से भी लिया, तो एज ए रिजल्ट if there are more RNA, that is equivalent to more of viral particles, and therefore you will have a lower CT value. The devices can be variable, And multiplexing you can do here also, like in BCR able, we are using multiplexing. So we have only BCR, we have only able, and we have BCR able, because we must keep the controls for the patient when we are doing, to be sure that we are looking at the chimeric gene and we are doing and in the background of that. So if you use multiplexing, then your fluorophore should be different. Everybody is familiar with Tachman strategy, right? Good. Now people have, now multiplexing as a limitation, you can maximum you can add 3 to 4 fluorophores. But supposing I say a disease fever, let's say I want to make a fever panel, point of care diagnostic for fever. Now there are so many pathogens which can cause fever, or even for pneumonia, or for sepsis in the hospital, because sepsis is very, very important. Now what do? I need 20 pathogens. So you can use a device which has come up like this. So they have, you put the sample in the middle, and then you have various chambers, right? And each chamber has only two set of primers. So the DNA will go into each chamber equally by the microfluidics, and each chamber has its own primers. So you know, you have labeled the these chambers are labeled from starting 1 to A to whatever B, C, D, E, F. It can have 6 or 8 chambers, and you can at least, if it is 8, you can determine 16 pathogen at a time, because it is multiplexing or duplex or triplex. So you can do it, multiple pathogens you can test in one go, one sample, and it will test for so many of these. This is very, very useful in diseases which have common overlapping symptoms. For example, fever chip, then you know STD, where I have been telling 20 to 20, any amount of, some are viruses, some are bacteria, some are fungus. So you can't have compatible primers for them. So it is better to use different vials with different set of primers. So, and not only this, now they have, or more beautifully, they have come out with devices which are these microfluidic devices on you know, various materials. One is of course microfluidic conventional device. Then they have come out with, I have not seen, but there are paper devices, paper based devices which have chambers like this. How they have partitioned, because you can't see these, but you have many of these are like that. I think I have one figure for that. So you can have many of these. So there are several pictures on the net you can see for that. And here in this first device that you see on the side, that is a handheld real-time PCR machine, which we had been working with one of the company. We have no expertise in electronics. They are doing all the electronics, developing all the, we are only validating the working of that machine with our diagnostic assay, with Tachman strategy, and this machine will be able, people will be able to use it for developing any diagnostic, because it has a chip on which you can put up to 5 microliter of real-time PCR, reaction mix lyophilized, and the assay that we have standardized, it is stable for almost 8 months in that lyophilized form. You seal it under nitrogen, and then you can distribute it. So that problem is also not here. In India, the temperature shoots up, so you can always transport it at room temperature, or at best at the 4 degrees, which will cut the cost a lot in transportation. Okay. So the idea is that, and the end user has to do nothing. Any technician can isolate the, cDNA, or who can prepare, he can isolate nucleic acids from the sample, and you have to put directly 5 microliter of the sample. Everything which is lying lyophilized will dissolve in that, and the reaction will start, because reaction is all programmed in the on the chip and in the machine. So machine will, it has software and hardware and everything will work accordingly. Okay. So you can, this is just the exponential phase and all that. So these are the single uniplex that we first standardized, and then we carried out the multiplexing, and you can see triplex, green, red and blue. So there were 3 different pathogens that were done, and this was these were the clinical samples, 30 clinical samples, their performance in this assay. So some of them are infected with both pathogen and some of them are infected with only 2, some are infected. Most of them were single infected or double infected, but very few were having all the 3 pathogens in it. Okay, so any questions from here? So I will then talk about the last bit of technique, that is now picking up a large way, although discovered about more than 10 years back, but only now people are becoming serious about its device, and you heard about it in the COVID also, LAMP assay. Okay, so this was first identified only for tuberculosis, but unfortunately amplification is so large that it would result in contaminating the whole lab. You do 10 days LAMP assay in your lab, and then the whole atmosphere is full of lamplicons only for that gene, and you start getting in negative control also, you will start getting the band. So you have to follow very strict conditions that this area is only for putting up the assay, and this area or outside is only for developing the gel or whatever you want to do. So it was not possible. When you are talking about low resource settings, then you can't have dedicated rooms for running the LAMP assay. So it never hit the market. But now people, after this microfluidics coming into and chip assays coming into diagnostic, people realize that it is very easy to even devise LAMP in a closed container. So if everything is there in the tube and I don't have to run any gels and only the color will change after the reaction, then it is very easy to say it is positive, it is negative. And bandi rehne do tube ko, you don't have to open the tube and just dispose it off. So there will be no spreading. Then there is another thing you can do is use the UDP. So, and you use an enzyme which will degrade all the DNA which has incorporated the UDP. Okay. So this will result in cleaning the whole system from the previous template that is floating around in the lab. So these 2 things had really helped in bringing back this LAMP assay into the picture, and it has lot of advantages. You really don't need a PCR machine. You have a water bath in the lab, 20 minutes of reaction, you can do it anywhere by even taking a beaker with that temperature. With 60 degrees, you can just maintain 60 degrees for 20 minutes without any problem, and you will get a this. So tuberculosis, why it became important? Because dot centers have to do, for each person who walks into dot centers, they have to do microscopic and then culture assay to be sure that they have tuberculosis. Okay, so LAMP assay can really help. So it is very simple, and why they call it is loop mediated amplification. So with every PCR, normally we get straight lines, we get DNA. Here it will loop back and it will keep on looping back, then it will act as a template. So you get lot of loops and like a cauliflower structure. And what happens in cauliflower leaves, one leaf ends and second leaf starts right before it is ending. The third leaf will start even before the second leaf is ending. Today you go and try to peel the cauliflower and you will see the leaves are overlapping each other.
Cauliflower or cabbage?
Cauliflower.
Okay.
Oh, sorry,
Cabbage you meant.
cabbage. Okay, cabbage. So because of that, you will have this overlapping structure. So you end up seeing lot of, and because of this kind of a structure looping mechanism, you get huge amount of lamplicons. But the only problem with this is the size is variable, whether it is one loop going into another loop, so the size is variable. And if you look at the gel, you see a smear. You don't get bands like in PCR. So one is always doubting whether there is a specificity or not. That is the only problem. Yes, what is your problem?
Ma'am, in the normal PCR, we are getting segments,
Hmm.
four-generic segments, and here we are getting loop. So what is making it happen this way? What's the
Because I said no, the primers fold back. So you have, but the specificity lies because you have at least two sets of primers for amplifying the same gene
in lamp?
in lamp.
That is
You can have three set of primers. So this figure explains little better. So if you look into F2 against a complementary structure to F2, then when amplification will occur, because it will anyway loop back to the F1. So you can see here F1C, there is so it is little, I will not be able to explain in such short time. So they have primer 1 and primer upstream of that is actually complementary to the other strand, and this is forward, genuine forward. So as a result it will form a loop because it will hybridize.
so the
Right,
for the next.
for the next one.
Okay, got it.
So it keeps on getting looped, and that is why it is called loop mediated amplification
This is not happening in the normal PCR.
in normal
There we
PC.
have straight
Only
two
straight
different
primers at the
primers.
two ends.
Okay,
Okay.
got it.
And because how many loops are being formed, so you can see in the bottom one A and B, so there can be F and F1C, so there is a 3' end and it will amplify, and here there is a new primer that is binding to F2C and F2, so that also will start. So you end up making lot of amplicons like that, and that is why you get this smear like structure. So people developed as the requirement was, so they didn't want to first run the aerosol because that will result in the forming aerosols of these lamplicons. They are called lamplicons instead of amplicons, and the whole lab gets contaminated. So they develop dyes. They will, if there is a negative sample, then it will be brown in color. If it is positive, then it will green in color. You put cyber green in that. Okay. So, but then people felt that its sensitivity can be increased and it can be made more user friendly if we spot a very small amount on a paper strip, and it will migrate and it will tell you whether if there is an antibody antigen reaction, then it will give you a control band and it will give you a pathogen band. And you can do, it is called lateral flow dipstick. So all you need to do is put either a drop or you just touch the strip to the lamp assay mix, and you will see it will move, and basically it is again on the strip you have antigen antibody. So test band, you can see here there is an antibody antigen reaction. So when the DNA comes here, it will interact, so you get a positive band, and there is a control band is also there, so you will have this bands are there. So you can see these two bands kind of a thing, control band and test band. So control band should always come, test band can come only in the positive sample but not in the negative sample. And here are some results from our lab where you can see those which are positive for tuberculosis. And you can see here intensity, in some cases test line is very dark, that means the person is heavily infected, for other it is light, and for some it is completely missing. So you can see this. So this was a very easy assay, and we could even tell how much we had loaded, one microliter or 2 microliter, and but we found even the tube assay was very reliable, because you could very easily see. So here is the comparison. So upper one is cyber green, different concentration. This was a turbidity assay, different concentration, from 1 picogram to it keeps on going to the 2 nanogram, or I think tube number 1 is 1 nanogram, 100 picograms, then 10 picograms. Tube number 4 is 1 picogram and 100 femtograms, like that, and then the rest, to find out the detection limit. Same is true for this. This is the gel picture where you see all the stream, and here we do the lamp assay, this lateral flow assay, to show that the sensitivity is slightly compromised when we do the test lateral flow assay, unless we put the whole amount. In this we are putting only 1/10 of the amount, because that is much the paper can take over. Okay, so point of care devices for protein analysis have started developing, but we still lack biomarkers, and therefore they will be, once identified, they will be good, faster, and the idea is to again develop lateral flow assay. There was a home test during COVID which was being utilized, and that was partially based on this strategy, where you had IgG and IgM protein detection on using the same lateral flow. And there you don't have to amplify anything, because you are looking for presence of the antigen in the blood and directly putting it on the sample, and then due to capillarity, when it moves, then it has its antibody already attached at the test line. And there were 2 test line 1 and 2, one for IgG and one for IgM, and you could find out whether the assay you were positive at home. So this was the home test that some people use. But the only problem was IgG and IgM was that the antigen reaction develops only after 4 to 5 days of infection. Okay, so some people in the early time, they would miss it, and then the doctor would tell them, but symptoms would not persist, so they will do it after another 2-3 days, and then they will test positive. But the problem is that by then they would have spread it to many other people. Okay, so the crucial aspect to consider in all these paper based assay or strip based assay is the surface characteristics and the capillarity. So in microfluidics, we know what kind of fluidic motion we are having, but when we do the paper, there was a cloth based microfluidics device also which came, and many new novel smart materials came up during that time. So depending upon, even for the paper, whether it has come for wood, cotton, flax, jute, bamboo, so each had their separate properties. Because their wetting properties altered, and their behavior of these fluids changed. So surface chemistry was very important, and porosity was very important. But people are coming out for these, and they are they are putting these for protein markers. They are doing a lysis buffer is put on the protein sample, on the paper or on this device, and then the proteins through porosity and due to capillary action, they move towards the site, and you can spot the antibodies for them and try to identify what all samples are what all antibodies are available and are present there. So, so that has given rise to 2D and 3D microfluidic strips where you can do photolithography. I'm sure many of you are familiar with that. And you can do plasma etching, wax printing, inkjet etching, and you can have many of these devices. So since we have some cancer C-reactive protein, so people are trying using that as a model system to test whether they are able to identify C-reactive protein using these similar strategy as we had seen in the microfluidics in the beginning. And you have gold nanoparticles, and you can hook them to these nanoparticles and try to develop these devices. Okay. So many more futuristic research, however, is required, because you need to handle, one thing is the small amount of the fluid should be used, because many a times it is not possible. I will give you an example. If you go to a leukemic patient and you want to take the blood, 2 ml of blood, for doing your genuine real-time PCR, so you need to isolate RNA and cDNA. It's very difficult. From leukemic patients to take another 2 ml of the blood, normally you will hardly get blood out of them. It is not that they have zero blood, but it is only whitish fluid. There's very low hemoglobin in there, and there is very, very little plasma in that. And it is really pathetic in some cases to take blood from children. There you can't even get 2 ml. You can hardly get 500 microliter from children, because some of the cancers are childhood leukemias, so there is huge problem. And even for other tests, you might have noticed that in pediatric section, many a times they take the blood from the foot of the child, because you cannot pierce here, the veins are too small. You can't take out the blood, and you will always hear them screaming to the top of their voice. Even for vaccination, they are scared of injections, and then taking out blood is very difficult. So the idea is we should have even less invasive, like if we can use saliva, if we can use nasal swabs, if we can use urine, then it will be very good. But then if we have to take blood, then it should be pinpricks or very small amount of blood, so that the patient is not harmed, or patient can give that much of blood. So minimum handling, small handling of the volumes, and that can only come with your microfluidics. It is very, very important, whether it is on paper or on the device. So we should go to femtomolar detection sensitivity from millimolar. We should have multiple marker panels. It should not be single panel, because the single protein or single PCR site or gene, because that can at times give us wrong results. Okay. And then of course low cost, long lasting material. That means they should not be, we should not be carrying them in -20, -80 stuff. And there should be ease and self-containment should be there, and robustness and accuracy. And so this is the ideal situation. So we have to work towards reaching these ideal situation, and the future lies with you people. You know where to go and what to contribute to the society in large. And thank you for your attention. I'm glad all of you came on the holiday to come for that. All right. I don't know like one specific ones, but lot of microfluidic devices are coming up now in market, and depending upon their intake, how much volume they can take, people are trying, but they are still exuberantly costly. So they have not come to daily, like real-time PCR machine that I have told you. It is portable, but they are not ready to sell it less than 5 lakhs right now, because they have put lot of money in their R&D. So unless they become equally competitive and less
Demand should
expensive,
be there,
exactly,
more of demand.
more demand, but that will happen gradually. And to the microfluidic device or the chip, it does not matter where from the sample is coming.
No, but for the convenience, like EVs are very high in urine and saliva sample, so it's easier.
Yes,
No
yes.
blood prick and nothing is
Yeah. So on the microfluidic device, you are going to put one preliminary extraction you will do. Whatever is there, like even if I take any body fluid, I probably will do a strong spin, collect the EBs and suspend it in the lysis buffer, because the DNA has to reach. So I have to lyse. If even if the protein is there, I have to put protein lysis buffer. So somewhere I have to lyse it, because macro structures of will not be able to penetrate the microfluid fluids of the, or the porosity will be, if it is that porous, then it will not be able to fractionate the material. But who knows, they might come out with better devices in future, because on chip lysis they are trying to do. Gene Expert for MTB sputum is very difficult to handle, but Gene Expert has devised a method by which you put directly the sputum first in the chip, the sputum will be lysed, the DNA will be extracted. You don't have to do anything. The machine will keep running for 30 minutes. They have the lysis buffer inbuilt and washing buffer, everything, and they have a kind of a gel sink in which all the fluid will go into that and get stored there, because in the end you are going to throw anyway that chip, and then only the DNA will move to the PCR chamber. So they have device, but it is their trade secret, it is not out. Nobody knows how to do it.
So, ma'am, in microfluidic devices, do we need to prepare our samples in advance, or they can be easily?
No, this I just told you in Gene Expert MTB, and now they are coming out for other infectious also diseases. They are coming out with that same device. I know they have couple of more in their catalog. But I don't know, like I showed you this paper based kind of or textile based devices. I don't know how much they can do it like that. Although in their picture they are showing that you put the protein extract, so but you have to isolate the extract and do the lysis, because then only the protein interactions will separate, or you put the cells and do lysis. So things have started moving in this direction, but they have not, everything has not come to the market yet. Many of them are actually still in R&D phase, but in advance. And the advantage of devices is, they don't take as much time as FDA drug approval, because they are in vitro. So they only have to come and show that their device is as sensitive or more sensitive than the existing one, and it is easier to use than the existing device. So that if they are able to demonstrate through their device, then it can be launched in the market. And if it is good, the marketing will take over. Right? So that way, those which are in R&D, we can hope that in the next 5 years we will have many such devices for diagnostics. Like liquid biopsy didn't come very many years back. It is quite recent, liquid biopsy, and it is already being done in India. Many these private hospitals are using liquid biopsy, so it is picking up fast. So I'm sure these devices will very soon get launched. Some of them I am told that they are in the this validation, clinical validation phase in USA and some in Europe. They are already in the validation phase, so they might hit the market very soon.
As for now, I have just seen one microfluidic device that was using Rajiv Gandhi for CTC actually, one microfluidic device. Other than very few market.
Yeah, and then MTB, you can say Gene Expert. Gene expert kits are there now, because once they have developed that device, now they are just adding more and more diagnostics to that. So, but we need many more, because they can't capture the whole market anyway, because इतना तो किसी भी कंपनी का मैन्युफैक्चरिंग रेट नहीं होता. We need lot of such devices. So, fine.
Thank you so much, ma'am, for your time. And as our honorable vice chancellor often says, that we have the national experts here who are the best in their fields, who have been delivering their decades of experience in just 1 or 2 hours. So we must thank ma'am for her time and